turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists podcast, the programme that brings you the biggest breakthroughs and talks to the major movers and shakers in the worlds of science, technology and medicine with me, Chris Smith. Coming up, a magic bullet for ovarian cancer, how a killer fungus is taking nature's fight to an invasive species, and the aftermath of Blue Origin's New Glenn rocket fireball failure. An ovarian cancer drug, which has been dubbed a biological missile, has just been made available on the UK's National Health Service. It's called mirvetuximab, and it delivers chemotherapy directly to cancerous tissue rather than the whole body. It's hoped that it will reduce side effects associated with some cancer drugs and will also help to prolong the lives of some people who have the disease by helping to treat disease that has become resistant to traditional treatment. I've been speaking with Dr. Raoul Miller, who ran the clinical trials at UCLH, about how ovarian cancer has traditionally been treated and what this new biological missile involves.
We usually treat with a drug called carboplatin, which actually works very well. But with time, the cancer becomes resistant to the carboplatin and other types of chemotherapy. So we call this platinum-resistant ovarian cancer. And it's really been a really challenging group of cancers to treat.
And how have you gone about trying to tackle that?
What we've done is we've looked at ovary cancers and we've looked at, do they have any markers or proteins that are unique to the cancer cells that you don't see anywhere else? And what we found is in ovarian cancer, particularly advanced ovarian cancer that was resistant to the treatment, a significant proportion of the cancers had a high expression of a folate receptor. So this is something that's expressed on the outside of the cancer cells, but we don't find that anywhere else. And that made that an attractive target or something to look for a treatment.
It's basically the cancer cells are waving a flag that you can see chemically, but how can you exploit that then to make the treatment more targeted?
So there's a class of drugs called antibodies, and these are molecules that attach to the flag that you described. So they stick themselves to the cancer cell. And if the marker or the flag is on the cancer cell and not anywhere else, then you're delivering that drug straight to the cancer cells. So what they've done is this technology whereby you combine an antibody with a chemotherapy molecule. So what you're doing is you're getting this molecule which homes to the cancer cell, it sticks onto the cancer cell, and then attached to it on the tail is this really potent chemotherapy molecule, and it delivers that into the cancer cell. So then you get the chemotherapy within the cancer cell. It damages the cell, causes the cancer cells to die, but it doesn't go anywhere else in the circulation. How that compares to normal chemotherapy is when we give people chemotherapy, normally it goes everywhere and then you get the side effects, the hair loss, the sickness. But this targeted chemotherapy just goes into the cancer. So you get lots of cancer cell death and much less side effects.
And does it bear fruit in that way? So when you actually subject this to rigorous clinical trial, do you see that being manifest and also solving the problem you set out to solve, which is when these become resistant to traditional chemotherapies, this begins to make the cancer respond again?
Yes. So the first question, do we see that? So in lab studies and pre-studies before we got to human clinical trials, we saw that that's where the drugs went. The drugs were delivered into the cancer cell. And then in practice, when we give these drugs to patients, we see a good result. So compared to standard chemotherapy, in many more patients, the cancer shrunk. So they had the tumors shrunk on the scan. So people feel better because they have less cancer there. And importantly, it translated into improved survival. We increased survival by 30%, which is the first time we've ever seen an improvement like that in this group of very hard to treat cancers.
It certainly sounds encouraging. The thing with cancers is that they are a moving target, as you alluded to, but also they're a mixed bag. Some cells do have these magic markers that you can see, some naturally won't. So are we really just kicking the can down the road a bit with this? Because all you'll do is select out cancer cells that don't have that flag you're using, and we're back to square one.
So that's a very interesting question. So for this particular drug, they needed quite high levels of expression. So we wanted at least 75% of the cancer cells to have a reasonable level of this flag. So that's the first thing to say. So most of the cancer cells will have this marker. But actually, with these drugs, we see quite a neat thing called the bystander, which is known as the bystander effect. When you get the chemotherapy in the cells that have the marker, it causes the cells to die. That chemotherapy spills out into the adjacent cells, which maybe don't have the marker, and causes them to die too. It's not a cure. We're not curing people with this, but we're able to control the cancer for a longer period of time than with conventional chemotherapy.
Given the benefits you've seen, is there a case then for not waiting until a person fails on all the existing treatments before you do this? Is there a case for doing this from the get-go and hitting it really hard with something extremely targeted like this? Does that buy you anything?
We're already on that, Chris. So there's trials ongoing with this drug and other drugs with other targets, which are perhaps present in more people with ovarian cancer right from the offset, after diagnosis and after surgery. Hopefully, over the next couple of years, we'll see some results from these big studies to see whether actually, if we use these drugs earlier on in the disease course, do we have a better outcome or a bigger improvement?
And how do the patients fare on them? Do they find them better and more tolerable?
Yeah, they do in general. So when you look at like the studies and what they say about quality of life, they do say it's better. But on a day-to-day basis, I think the most important factor is that the patients do tolerate these treatments well. They're not without their side effects, and some of those side effects can be slightly different from what we see with chemotherapy. For example, sometimes you can get some irritation in the eye and some blurred vision and have to use eye drops. But in general, these treatments are better managed. Patients get on with them better than they do with the chemotherapy that we use, and particularly in this group, in this setting, the standard treatment that we used was weekly chemotherapy, and this treatment is 3 weekly. So that's a huge gain in terms of time back for patients and time away from the hospital, which is really important.
Brilliant news. Rowan Miller there at UCLH. For decades, astronomers believed that black holes grew from a collapsed massive star, and that meant that galaxies inevitably already existed by the time that was happening. But recent findings from the University of Cambridge suggest that this might not actually be the case. Astronomers have been able to measure the mass of a black hole appearing as a distant little red dot dating from when the universe was just 700 million years old. This black hole is entirely on its own and absolutely enormous, far too big to have come from a collapsing star. So it suggests that some black holes formed first and then gathered a galaxy and a cadre of stars around themselves. Here's Ignace Georgibalis.
So the backdrop to this and all current studies of black holes in the early universe was that we still really don't know where the supermassive black holes that are at the centers of galaxies, for example, the Milky Way has a black hole that weighs as much as 6 million suns, or 4 million suns thereabouts. And we still don't know where these black holes came from. So one of the main goals of the James Webb Space Telescope was to actually observe black holes that existed when the universe was very young, a fraction of its current age, and to then try to figure out where the current supermassive black holes came from.
How far back in time are you looking with this study?
So about 12,800 million years back. So we're looking at the universe when it was just 700 million years old.
How do you know you're that far back in time?
So this requires a concept called redshift. So essentially, what was noticed back in the 20th century was that distant galaxies appear to recede from us, and that recession velocity is proportional to its distance. So that was the first hint that the universe is expanding. And then basically, if you can measure the redshift that's caused by that recession velocity, then you can figure out how far away something is. And since light is traveling at a finite speed, for example, it takes light 8 minutes to reach Earth from the sun. Well, it takes well over 10 billion years to reach us from very distant sources. And since we know how old the universe is now, we can then know how old the universe was when the light was first emitted.
And what are you imaging from that far back?
So for this particular study, we were focusing on a very interesting black hole that was already spotted in the early universe. And the interesting thing about it, that people who first spotted it notice, is that it does not seem to have much of a galaxy around it. So normally you would expect to see, well, that far back, you would still see a fuzzy blob. But in this case, the object appeared to be a perfect point source.
And how do you know it's a black hole at all?
Right. So the main clue was in the spectrum of the object. So we were looking at the colors of light coming from that object, and we saw that the colors effectively that correspond to the emission of hydrogen show signs of the hydrogen gas moving at very high speeds, which is generally only seen when the gas is interacting with a nearby black hole and swirling around in its powerful gravitational field.
Does the speed at which all that gas is moving, you can use that to work out, therefore, how big the entity is?
Yes, essentially that's the meat of the study is we use the speed at which the hydrogen gas is moving in that system to directly work out the mass of the black hole.
And what is its
And
mass, do you think?
50 million suns.
And is that big
and
in the black hole continuum? Would that be a big black hole?
It's about 10 times bigger than the Milky Way's black hole, but there are larger black holes seen in the early universe.
Now, that's much bigger than any star, as far as we know, could be, isn't it? If you had something 50
Yes.
million times bigger than the sun. And our current idea about the origins of black holes is big stars, they get to the end of their life, they explode and then implode into a black hole. So does this mean there's another way that black holes can form without a star blowing itself up?
Yes, this has been suspected when those other large black holes were seen at similarly early times. So what separates this black hole from the others is that it does not seem to have much of a galaxy around it. When we look at the velocities of hydrogen gas, they look like the velocities of the planets in the solar system, where gas further away moves slower. So that suggests that all the mass is concentrated in the black hole. Whereas if you look at a normal galaxy, most of the mass is not in the black hole.
So where did it come from then? What do you think, or theoretically, hypothetically, how could an entity like this form?
So there are two ways that we discuss that seem to sort of survive this measurement. So one is that under certain conditions, if you have a gas cloud, you can have the cloud collapse directly into a black hole that would then have less to grow to reach a very large size, and you could still have not many stars around it. So that's called a direct collapse black hole model. The other one that is even more exotic is that under some currently very theoretical physics models that talk about what happened when the universe was just born predict that you will have black holes that have formed all the way back then. So potentially those what's called primordial black holes could have made this object as well.
Is that because during its younger phases, the universe, there would have been greater densities of gas, so the likelihood of them being able to fall in together to produce a sufficiently big mass that could then lead to a black hole was greater in likelihood than it would be, say, today?
Yes, that operates also for the direct collapse black hole ideas. For the primordial black holes, people say that, when the universe was born, the gas was not evenly distributed in it, because if it was, then no galaxies would have ever formed. So there were clumps that were denser, and then under some models of how those clumps were generated when the universe was born, some of them would have been too dense to form anything else but a black hole.
Does this suggest then that one possibility is that the early universe gets a whole bunch of big black holes and they act as a sort of nidus around which galaxies then form? Is that how we think you end up with a situation as we see today with galaxies like the Milky Way with massive black holes sitting in their centres?
That is one possible mechanism, and it likely does operate. It's not just based on this measurement. As I've mentioned, there's currently quite a few other teams that, they haven't done the same observations that we have, but other channels also point to this being the case.
It just completely blows my mind to think that we are seeing all the way billions of years back to a universe that was just 700 million years old. It's absolutely incredible. Ignas Georgbalis there. That discovery just out in Nature.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound. Perfect music for audio and video productions.
This is the Naked Scientist podcast with me, Chris Smith. In a minute, the rocket that exploded on the launch pad with the destructive power verging on that of a nuclear weapon. What has that done to NASA's lunar ambitions? Before that, though, to the ground under our feet. During World War II, the UK was invaded not by marauding German forces, but by a new moss species called the heath star moss. Since that time, it has inexorably encroached across the environment, turfing out native plant species as it grows. You can find it everywhere, from bare soil and rotting tree stumps to covering cracks in tarmac and even on thatched roofs. But 4 years ago, while he was on the Isle of Wight, George Greif, who is now based at the John Innes Centre in Norfolk, noticed patches of dead moss on a cliffside, and he took some samples away. His post-mortem revealed that the cause of death was a killer fungus that appears to have adapted from life on a native moss species to take on the invader, as Rachel Ralph has been hearing.
So we found a moss dieback fungus. So this is a fungus that basically kills an invasive species of moss. This moss is called the heath star moss or tank moss. It's an invasive species that arrived in the UK in around about 1941, they think, on military equipment. It's kind of really spread insanely since then, occupying a lot of different habitats across the country. It's a generalist species, so it's able to grow in a lot of different places, but it's particularly bad on heathland and sand dune ecosystems.
When did you first observe this fungus infecting the moss?
So it's quite a long story. I think I'd been seeing it and collecting it since about 2017, but it was quite a tricky fungus to identify, so I kept kind of seeing it and throwing it away. But about 4-ish years ago now, I started finding a lot of it and noticing a lot more of it and kind of looking for it specifically, and realized it really is kind of basically in every heathland, at least, that I was looking in, and started to study it in detail then.
And when you examine the infected moss, what do you actually see? Does the moss change colour, or is there any patterning at all?
Yeah, so this actually forms, well, this term seems to be liked in the news article, fairy rings of death. So this is like brown patches of kind of holes in the moss, which otherwise forms these very lush, dark green, thick mats on the ground. It doesn't allow anything else to grow. But the fungus is basically forming these holes in it, which are like these rings that grow outwards. They're brown, the moss is dying. There's like a white fluff on the tops of the leaves of the moss in the frontiers of the infection as it spreads outwards. So it's quite striking, actually, and you can see it in well-developed fungal populations, at least, quite easily with the naked eye.
How do you actually know it was the fungus killing the moss? What kinds of methods did you use to determine that?
Yeah, so initially we kind of just, take a sample, take it home, look at it under the microscope, under a compound microscope, and see what's going on, and then kind of identified it was a fungus. But as I mentioned before, it's quite a tricky fungus to identify, and it doesn't have many features that allow it to be identified just by its appearance. So actually, the thing that really helped a lot with this was DNA sequencing. So I was able to take some of the fungus, put it in a tube, sequence it, and that revealed that it's actually part of this, in this genus called Bryoscyphus, which is a group of moss-killing fungi. So that kind of helped. And then, eventually started connecting with other people who'd found the fungus in France and the Netherlands, and we really put it all together and sequenced everything, found it was all the same, and put a description together.
Do you know exactly how the fungus is killing the moss?
Yeah, so that's a good question. The fungus seems to be growing inside the moss cells, so it kills it from the inside. It also grows on the surface of the cells, so it seems to produce structures on the outside as well. So it's really kind of attacking it in both ways, probably with chemicals and physically with its mycelium as well.
So of course this fungus is infecting the invasive heath star moss species, but do we need to be worried that the fungus can also infect other plant species that aren't causing a problem to the environment?
Yeah, so the fungus doesn't grow on, well, you'll be relieved to hear it doesn't infect humans, doesn't infect animals, it doesn't infect other plants as far as we know, except for one other moss, which is a native moss species related to the heath star moss. However, it doesn't cause as severe symptoms in the native species that we've observed so far. So that's why we think maybe the fungus is native and was growing on that moss, just happily living its life. And then when the invasive species came, it hopped onto the invasive species. That's one idea. It could also be the other way around, which would be a little less thrilling.
Yeah, that would be quite concerning. Do we also need to be concerned that maybe some of these mosses have a mutation that protects them against the fungus, and then they spread their spores, then suddenly we have both an invasive and a resistant moss species growing?
Yeah, I guess it is a concern, but you've got to realize like the moss is reproducing sort of the whole time and creating, it has quite a lot of genetic diversity that it is constantly adapting and in theory could become resistant to the fungus. But on the other hand, the fungus is doing it as well. So I think what would probably happen is one of these kind of co-evolutionary kind of arms races where if the moss changes, the fungus will change and vice versa, and they'll keep following each other. So I think the more likely thing we'd have to worry about is if the fungus is able to spill over and infect more vulnerable native mosses that are related to the heath star moss, but not the same one.
And is that what you're planning to look at in the future?
Yeah, so I've recently moved from working in the kind of museum sector where we're kind of using museum specimens like a kind of time machine to look in the past to see whether the fungus was on mosses before the star moss or the tank moss arrived in the 1940s. So we have these specimens and we can look. It's quite difficult and time consuming. We've given that a try. What I'm doing now is kind of working in the lab and trying to see how many different species of moss related to the heath star moss are susceptible to the fungus, try and work out its mechanisms of infection and learn a bit more about it. We can sequence genomes. There's quite a lot of exciting and kind of cutting edge science we can do on it.
It's an interesting story, that, isn't it? George Greif, and he carried out that research at the National Museum of Wales in Cardiff. Blue Origin officials have been assessing the damage caused after its New Glenn rocket dramatically exploded in Florida with a force verging on that produced by a nuclear weapon. The fireball occurred as Jeff Bezos' company was carrying out tests in the run-up to using the rocket to launch a batch of satellites for Amazon. Space scientist and author David Whitehouse has been telling me about it and the impact that it will inevitably have on NASA's lunar aspirations.
We're not quite sure what happened. I mean, we know there was an explosion after the main engines were firing in a test. So they'd already ignited the main engines. But what happened subsequently to cause this enormous fireball, we're not quite sure. But the thing which struck me is that the energy in this fireball was equivalent to about 1/10 of a Hiroshima nuclear detonation. It was an enormous explosion, and it shows you how much energy you have to use to get something into space, because that explosion released the energy in a few seconds, and it's completely wrecked the launch pad.
Yeah, I was going to say, it focuses your mind when you think about that amount of energy being unleashed, all in a fairly small space.
Yes.
So yes, there was fallout for the launch pad then.
Oh, the launch pad has been completely wrecked. Launch pads aren't just pads of concrete. They are highly complex pieces of infrastructure. They have lots of tubing, lots of pipes going in, bringing in fuel, different types of fuel, bringing in water, taking water away, the electrical components, damage beyond repair. The blast radius of damage extended more than a mile. So this is a major problem with getting this launch pad back together again. And the only comparison we've got is what happened to SpaceX 10 years ago when a Falcon rocket exploded. And that was a different type of explosion in the sense that it occurred at the top of the rocket and not at the bottom. But it took them a year to get the pad back straight. Some people have said there is a golden lining in this, in the sense that the pad which was destroyed was a fairly old-fashioned pad, and it was modified to deal with New Glenn. Now they've got a chance to really upgrade the pad so that in the future, once they get over this, they could perhaps have easier, swifter launch rates. But no doubt at all, it is a major disaster for Jeff Bezos and for the American space effort to get back to the moon.
Was there anything on that rocket? Because you said it was a test firing. Were they just lighting the engines, or was this a rocket that was going to go somewhere and going to do something?
It was going to go somewhere the following week. It was a test. It was a test of the engines. Nobody expected a problem, obviously. Fortunately, the satellites, the communication satellites that were to have been launched the following week were not installed. So that would have been an additional disaster if they had been destroyed. Now, those satellites have to find another route into space, but at least they weren't completely obliterated, which is what would have happened.
Didn't the Russians have a problem with one of theirs for a similar sort of situation? We spoke about it earlier in the year.
The Russian launch pads are simpler, and they seem to have a reasonably straightforward time, although it took them many, many months to get the thing back together again. But what Jeff Bezos got is a very complicated task, because his New Glenn rocket is central to getting America back to the moon. And the delay in that of at least a year gives timetable problems, but also Bezos political problems, because what he said was when he wanted to get the pad back working later this year, when I think people believe it's going to be middle of next year, is that he doesn't want talk of the satellites and the lunar craft that New Glenn was to have launched. He doesn't want anybody to say, well, let's give them to SpaceX, modify them and launch them on SpaceX. He doesn't want people to go down that avenue.
What role does this play in NASA's moon ambitions?
A central role. I think people realized a few years ago, when they were giving so much to SpaceX for the lunar return, that they did so because they said Elon Musk delivers. But then I think they realized that it might not be as straightforward as that. So they split it between SpaceX and Blue Origin. And what the New Glenn was going to launch was the Blue Moon lander, which is the central template for landing cargo on the moon. And that was to have its first flight later this year, where it was to put down a prototype Blue Moon lander at the South Pole. Now, this Blue Moon lander is an important bit of kit. It's much smaller than the Starship, which SpaceX wants to land on the moon. It's much more like an upgraded, much larger Apollo lander. And it's a start of a series of Blue Moon landers of various degrees of increasing complexity, leading up to something that could carry people to the moon. So it's vital to start the Blue Moon lander production line and landing on the moon in order to get equipment to the moon that people have already paid for, in order to develop the technology to put people on the moon in the next few years. And the timetable for that was pretty tight, because NASA wants to upgrade its cadence to get to the moon, starting very intensively towards the end of this year. And of course, now the Blue Moon lander cannot go for at least another year. That, in the short term, has a major effect. It had in a couple of years a contract to launch 2 rovers to the moon. And Blue Origin has said, well, actually, in a couple of years, we'll be back and running, and that should be all right. It's the next 18 months that is the problem, in terms of the fact that New Glenn is delayed, the Blue Moon lander is delayed. SpaceX is having problems with its Starship, because that's not coming on as quickly as people thought. So this is a major setback, just at the time when NASA wanted to say, okay, we're going faster now. We're sending more stuff to the moon. We really have to build up to the landing.
David Whitehouse there. Well, that's it for today. Tune in on Tuesday, though, where as the world reaches football fever pitch, we're going to be taking a closer look at the FIFA World Cup. It is reportedly the world's most watched and most popular sporting event. But is the competition more of a science than an art? That is what we will find out. I'm Chris Smith. Thank you for listening. If you'd like to get in touch with me in the meantime, it's chris@thenakedscientist.com. Until then, though, thank you for your company and goodbye.